Hi again, everyone. Today we're going to start into a two-part series on muscles, muscle tissue, and the overall musculature of the body in terms of where muscles, the names of muscles, as well as basic function. Muscles have basic, shall we say, physiological characteristics. They are excitable like neurons. They can undergo what is known as an action potential. That is to say, the membrane surrounding the muscle cell can undergo a voltage shift that we call an action potential. We all know that muscles are contractile. That is to say, they can generate force by shortening, but muscles are also extensible. You can stretch them. And they're also elastic, meaning that they can bounce back to their previous shape when stretched. The two primary characteristics that are most important are excitable and contractile, but these other ones do play a role in terms of maintenance and health and so forth. Based on those physiological characteristics, we can then extend into functions. Certainly, we generate movement with muscle. That is locomotion, manipulation, like walking, or smooth muscle function, like moving material through the digestive tract or the cardiac muscle pumping blood. But we also have muscles that primarily stay in relative contraction or can alternate contraction and relaxation in order to maintain certain things like posture or positioning in three dimensions. So stabilizing, a given body position, if you will, rather than movement. It can also stabilize joints. So these 2 and 3 are sort of similar in that way, but stabilizing joints is to prevent inappropriate articulation of a joint rather than, say, maintaining posture and body position. Fourthly, as they undergo metabolic activity in terms of contraction, they will make ATP, and that process can generate a fair amount of heat. So it is shall we say, a thermogenic tissue, if you will. And then lastly, they provide force for venous return and lymphatic vessel flow. We call that general process, we call that bulk flow. So when the cardiac muscle moves blood through the arteries and the muscles move blood through the veins and or the lymphatics, that's a bulk flow process caused by muscular contraction. Throughout the body, we have three general types of muscle tissue, though within that muscle tissue, there are a number of subdivisions. But these are the three main divisions of muscle tissue, almost at a tissue level. We have skeletal muscle, also known as voluntary. We have cardiac muscle, which is generally involuntary. And then we have smooth muscle, which is categorically involuntary, though you can induce it in certain ways. Like, for instance, swallowing starts off with a voluntary muscle, but then triggers an involuntary peristaltic wave down your esophagus. Whenever we see myo or MYS or sarco, those are all different prefixes regarding to something having to do with the muscle. Like, for instance, muscles for a long time were so specialized and different that there was actually a whole field of physiology that basically invented a lot of their own words. Like, for instance, cytoplasm in a muscle cell is called sarcoplasm. And that's because there were basically two parallel fields of cell biology at one time. And when we talk about skeletal and smooth muscle, we really are referring to what we call muscle fibers. Cardiac, those are unicellular and so forth. And so for whatever reason, even though I don't necessarily agree with it, we don't normally call cardiac muscle cells fibers, only the skeletal and smooth muscle by virtue of their generally elongated morphology. Cardiacs are more branched, they're not quite as long, and that's why they just haven't gotten the muscle fiber designation. When we do a side-by-side comparison of skeletal, cardiac, and smooth muscle, the first major difference is, where do we find these muscles in the body? With skeletal muscle, that's easy. You find these things attached via tendons to bones, with one exception, and that is because we can generate complex facial expressions. Maybe we ought to include one of the roles of skeletal muscle as communication, in fact. Well, let's not worry about that too much, but that's how we can generate complex facial expressions, is because we have some skeletal muscles that are attached to the skin in our faces. Cardiac muscle you find exclusively in the heart. And in some ways, cardiac muscle is kind of this in-between of smooth and skeletal muscle. It's striated like skeletal muscle, but it's involuntary, sort of like smooth is. And it has some electrical characteristics that resemble both skeletal and smooth muscle. But in any case, we find the cardiac muscle exclusively in the myocardium, okay, in the walls of the heart organ. Smooth muscle we find all over the place, and it has the widest diversity of any of the three muscle types because we find it in different organ systems. We can find it in the eyes, we can find it in our airways and arteries, we certainly find it in our digestive tract, visceral organs, the stomach, small intestine, large intestine. And so you do see lots of diversity. Even the erector pili muscles, which are found in your skin that give you goosebumps, are smooth muscles. So lots and lots of diversity there. In terms of cell shape, skeletal muscles are huge. And that's because what happens is when they are forming, the precursor cells, the myogenic stem cells, if you will, fuse together. Now that solves a problem, and it solves a problem of what we call uniform contraction. You do not want asynchronous contraction within a given skeletal muscle, because that'll lead to changes in direction and so forth. So we want a continuous, smooth, unidirectional contraction in any given muscle group. And so how skeletal muscle cells have solved that is they've fused together, and that's why they're multinucleate. And that's why they're really, really big and long, is because they have fused to form this long, gigantic cell. Cardiac muscle solves that problem that I just mentioned a different way, using gap junctions, and you can find them in these intercalated discs. You see these lines right here where my pointer is? Those are called intercalated discs, and those link one cardiac contractile muscle to another. And so there are gap junctions in there that allow for sharing of ions and allows for more uniform contraction. And cardiac muscles don't fuse. They're uninucleate. They do not fuse like the skeletal muscles do. They are striated like skeletal muscles are, but they also have this branched type morphology. They form these branches and they communicate with several other cells via those gap junctions. And so that's kind of solving the problem a different way than what we do in the skeletal muscle with fusing the cells. Smooth muscle, uninucleate, but you can't see any striations. That's why we call it smooth muscle in the first place, is because you don't see, it's a little tricky to see the striations in this heart muscle slide, but if you look closely enough in a microscopic slide, you will see striations. Smooth muscle is just that, it's smooth, and that's because the sarcomeres are not aligned in the same directions. Yes, this fiber will shorten, but it will shorten like a scrunchie rather than like a piston. Okay, so these are uninucleate, no striations, and we do have a fair amount of diversity when it comes to smooth muscle in terms of how they are stimulated. When we dig a little deeper when it comes to skeletal versus cardiac muscle cells, yes, they're both striated. They both contain regimented sarcomeres. That's why you see those striations. But the manner and how skeletal and cardiac muscle cells contract is very, very different. Skeletal muscles, by and large, it doesn't seem like this. We don't notice this, but most of our skeletal muscles are at rest most of the time. We contract them for relatively short periods of time or repetitively, but again, short periods of time. So depending on the muscle fiber type, we have fast twitch and slow twitch. You may have more or less mitochondria. Fast twitch fibers tend to have less mitochondria because they're using glycolysis, which is fast. Cardiac muscle, on the other hand, is very, very repetitive. It cannot afford to fatigue. If you fatigue, you lose effectiveness, and if you lose effectiveness, you stop pumping blood. So mitochondria are very, very rich in cardiac fibers, and it's in cardiac fibers cannot function anaerobically. They have to use oxygen. That's why you have lots of mitochondria. That's why you have myoglobin. Myoglobin is like hemoglobin, only it stores oxygen in muscle fiber. And you also have lots of glycogen, which is just polymerized glucose. We hear about glycogen a lot in exercise science. It is just a stored polymer of glucose. And so you got a lot of that in cardiac because, again, cardiac cannot afford to fatigue, at least not very much, right? Now, as I mentioned, skeletal muscle, you have aerobic and anaerobic. Aerobic tends to be more slow twitch fibers, and anaerobic is more glycolysis-mediated ATP production. Whereas, again, skeletal muscle is only aerobic. That's why it doesn't have a sense of humor. That's why we have heart attacks. All right, we only have, a few minutes if there's no blood flow there to get that blood flowing, or that heart tissue is going to die. Same thing with neurons as well. Skeletal muscle, there is some involuntary occasionally, but mostly it's voluntary. And cardiac is almost exclusively involuntary. Now, it doesn't mean you can't willfully change heart rate by doing behaviors, but it doesn't really need any input from the nervous system. It will beat on its own, even if you take a heart out of somebody else and put it into a recipient patient. It is responsive to hormones from the autonomic nervous system, and a normal heart that you're born with does have a little bit of innervation from the autonomic nervous system, but it's not essential. It's generally an autonomously beating, contracting organ. Skeletal muscle has individual motor units that you can recruit. That's one of the ways why you can have differential strength of skeletal muscle contraction, depending on how many motor units you recruit simultaneously. Now think about yourself grabbing a big old barbell and trying to move it. You'll grab it and you'll grimace. You'll move all your muscles in your face and your body and so forth. That's because your brain is literally trying to recruit every muscle fiber it can find. And that's and every motor unit. A motor unit is defined as a neuron and how many muscle fibers it innervates. You have big giant motor units down in your legs, which is why you can't sign a pen with your legs. And you have little tiny motor units in your fingers and in your face and so forth. Now, cardiac muscle basically functions as a single unit, and that brings back the importance of those intercalated discs. As you initiate a depolarization wave, it spreads through the tissue like a wave. We want to have this coordinated single unit type contraction. It's not all the fibers are contracting at once, but they are contracting in a wave-like manner. All right, a wave-like manner that involves a unit level coordination. Muscles can be repetitive. Skeletal muscles can be repetitively stimulated. That's why they're refractory period. That means that's the period that you could stimulate again and you get no response. Refractory means that it is unresponsive during that time. Okay, so if you fire, let's say you fire 2 pulses within half a millisecond, the skeletal muscle could not differentiate between those two. It would only fire off once. Now, cardiac muscle has this extremely long refractory period because we don't want what we can achieve in skeletal muscle known as tetanus. If you keep repetitively stimulating a skeletal muscle, it will go to, depending on how fast you're stimulating it, it will go to maximal tension and sustain it. We call that tetanus. Yes, there is a disease called tetanus, and that's because it stimulates skeletal muscle to the point that the body gets super rigid, extremely painful. All right. Well, we don't want that in cardiac muscle because tetanus in cardiac muscle is the same as asystole, okay? Is the same effect. If your muscles in your heart are just squeezing all the time, they're not moving any blood. So we can't afford that. We'll die. So Cardiac muscle has a long quarter of a second. That's 250 milliseconds. 1,000 milliseconds would be a full second. So we're talking about a full quarter of a second that you can't stimulate the heart again to contraction. And that implies that there's about, the maximal heart rate that you could ever get out of a human heart would be 240. You think take 60 and multiply it by 4, okay? It'd be 240. So whenever we get into 180, 190, that's kind of, people are starting to worry about, maybe the heartbeat's too fast, okay? Long, okay, they're long, they're multinucleated, they're big giant cylinders. Cardiac muscle short branched, intercalated discs that have these gap junctions and desmosomes to hold them together so they don't rip apart, so forth and so forth. So there are 3 main points in this slide. We don't want to get too far afield here. There's a little bit of detail here that maybe isn't quite as important for this level. But one thing that we should know is that there's good blood flow. And that's because the skeletal muscle is among the most energy consuming tissues. It's the most metabolically active tissues in the body. And when we begin exercise, blood flow increases massively to muscle. To the tune of 12-fold or more blood flow increase to skeletal muscle. That's one of the reasons why they say don't go swimming after you eat, because when you eat, you're basically dilating blood vessels to your digestive tract, and then you turn around and go swimming, which is a highly energetic activity, and now you've got this basically this competition between your digestive tract and your skeletal muscles. So there's good blood flow there. Now we also have connections. We have to connect this muscle to other tissues, and you can do it a number of different ways. Most common is via tendon. Okay, sometimes you can connect to at the periosteum, but most of the time we're talking about skeletal muscle to tendon. All right, and then that tendon is actually connective tissue. It's dense and irregular, and depending on where it connects to in the muscle, whether it's deep down inside or it's on the skeletal fiber surface, okay, we call them the myofibril, or it's even outside, it's a little bit further out, then that's not as important as the fact that it's dense and irregular connective tissue. Just a little bit more about skeletal muscle fibers. All right. Now, if you look here at this myofibril, what you have with a myofibril is a series of sarcomeres. Okay. A myofibril is several sarcomeres in series. Sarcomeres are the actin and myosin contractile units, and that's why you see this striation, if you will. It's due to the lining up of the sarcomeres. Now, skeletal muscle really only has a couple of seconds worth of ATP. It's not a big ATP storage cell. And so it has multiple ways of obtaining additional ATP. And one of the most common, and this is like, for instance, let's say for a moment you forgot something out in your car. And so you get up from your chair and you go out your front door and you go out to your car. Let's say it takes you 30 to 60 seconds to do that. The first 2 or 3 seconds, you might just use ATP, but after that first couple of seconds, your body, your skeletal muscle has to begin generating more ATP. It cannot afford to run out of ATP. There are so many reasons why, okay? Whole bunch of reasons why. In fact, if you look at fatigued skeletal muscle, you don't see a major drop in ATP. That's because the cell will die if it doesn't have ATP. So we've got to start generating ATP in a hurry. And how we do that is we use this enzyme. It's called creatine kinase. Say the word, don't just go kinase, okay? It is creatine kinase. And creatine kinase can remove phosphate from creatine phosphate and transfer it onto ADP to make it ATP. Or when you are resting, creatine kinase will take the phosphate off ATP and transfer it to creatine to make creatine phosphate. Meanwhile, since you're not working very hard, the cell will regenerate that ATP using oxidative phosphorylation. So you make creatine phosphate when you're at rest, and then you use the same enzyme to transfer that phosphate back onto ATP when you're at work for the first 10 to 60 seconds. So if you're running a 400, for instance, that's mostly creatine phosphate. All right. So there is a clinical correlation. If people have trouble with creatine kinase, they do see this condition, which is this is called rhabdomyolysis, which is myolysis, which means muscle destruction. Okay. And that seems to be involved with something going wrong with creatine kinase. One more thing, I've already mentioned about myoglobin in a previous slide. It's like hemoglobin. It does have a heme functional group in it. It's got an iron with a carbon ring around it. And it can hold on to oxygen to provide a little more oxygen so that you can generate ATP using oxygen, even if the blood flow isn't entirely there. So again, the big pieces of this, creatine kinase, all right, allows us to utilize creatine phosphate as an ATP source during short bursts of activity that last longer than just a couple of seconds. And myoglobin, that was the other main one there too. When we stimulate a muscle fiber, we want that myofibril to contract uniformly. We don't want one half of it contracting and not the other. I'll do a demonstration in class on this. If you only have half of your muscle fiber firing, that would cause a deviation towards the side of contraction. And that is extremely painful and damaging to the myofibril. So we don't want to do that. So you have a couple of structures. One of them is known as a T-tubule. This is a specialized type of cell membrane that allows for uniform spreading of the action potential across the myofibril. You also have these cisterns, which are part of the endoplasmic reticulum, which in the muscle we call sarcoplasmic reticulum. And these cisterns contain calcium. The SR, the sarcoplasmic reticulum, contains calcium. So the T-tubules are about the excitation. The T-tubules will trigger calcium release from the sarcoplasmic reticulum, which then leads to contraction. If you can see a sarcomere at the microscopic level, depending on where you would do a section through that muscle fiber, you would see over an alternating band or pattern of what we call thick filaments, which are myosin, which are shown here in red, and thin filaments, which are actin, that are shown in blue. The way that skeletal muscle fiber contracts is that the thick and thin filaments progressively overlap more and more. All right, they progressively overlap more and more. And so that sliding filament theory is the story behind skeletal muscle and cardiac muscle contraction. It's called the sliding filament theory. So those sliding filaments, we have thin filaments made up of actin. That's the primary component. That's the blue spheres there. Actin can form monomers. Those little blue spheres are monomers of actin, or they can be polymerized into this helical structure. Okay, that's called filamentous actin. Okay, so you have the actin in blue. And then wrapped around that helix is another complex of two proteins. One is called troponin, which is a calcium sensor protein, and the other is called tropomyosin. All right. So one more little teeny tiny thing here, and that is the actin polymers. You can see there's a little black dot on each one of those. That black dot right now in this picture is covered up by the tropomyosin. The tropomyosin is that orange line, and the troponin is there in yellow. Again, troponin is a calcium sensor protein. The tropomyosin is covering up a part of the actin that can bind myosin very, very tightly. All right. It will, when uncovered, will bind and grab onto a myosin head. We call that a cross bridge. Okay, and that's the thin filament. It is primarily actin, troponin, and tropomyosin. The thick filament, on the other hand, is all myosin. Okay, it's all myosin, and myosin has two major, or shall we say 2.5 major structures. You have the thick filament tail, right? Sometimes known as the heavy chain. All right. This is the thick filament tail, and you can see the thick filament's actually composed of a whole bunch of those tails. You have a hinge region, that's kind of the half, if you will. And then you have this catalytic head. Okay, this is the part that will bind to actin when that high affinity site is revealed. And this is also where we will hydrolyze ATP. This is the catalytic part of this. This heavy chain, this part right here, is more for binding to other myosins and strength in that thick filament. So we have actin and myosin. You have troponin and tropomyosin. Okay, we hopefully got there. A couple more things that we want you to know about. Okay, just a few more. And again, the key is that you don't have to know everything about these. Just write down 1 or 2 words for each of these. Okay, one is called titin. Okay, titin, it's the largest protein we know of. Like, it's titanic. It's huge. If you were to spell out the chemical name of titin, it would be over 1 million letters. It's huge, massive. Titin is an elastic component. So you know about isometric contractions. You know, when you like push against a wall or something and the wall doesn't move, but the muscles are still generating force. That's because the force is conferred onto this elastic filament titin, and it will absorb that tension energy. So titin is an elastic element in muscle fiber and prevents the muscle fiber from ripping itself apart. All right. Dystrophin helps link thin filaments. It helps relink the actin filaments. And again, dystrophin is part of the strength element of, in terms of it, when you contract. People who have muscular dystrophy, like Duchenne's muscular dystrophy, that's what DMD stands for. When they undergo normal levels of muscular activity, their muscle fibers begin rupturing in a way that is very, very similar to what happens in people who don't have DMD, but at extremely high levels of physical activity. Okay, so if where you're when you're doing really, really vigorous exercise, you actually can begin doing damage to your muscles. Have you ever heard about like throwing your arm out in baseball? Pitchers. That's why pitchers need rest. In the professional baseball, is because they're throwing so hard they're actually damaging the muscles, and they need a couple of days of rest. Well, with Duchenne's muscular dystrophy, that happens under just getting out of bed. All right, so it links thin filaments and it helps prevent cell rupture. The main one here is nebulin. Yes, there are these other ones. I'm not going to worry about these things. But if you see a weird one and it's talking about muscle, really they're just there to kind of make sure that the muscle fiber stays aligned. Okay. Nebulin is the main one. They call it nebulin, like it was nebulous what its activity was. It means that you didn't know what it was doing, and the name stuck. So they help hold the alignment of the sarcomere, these other proteins, because again, asymmetric contraction will cause rupture and destruction. And lastly, the Z disc, which is in between sarcomeres. If you look at some previous pictures, you'll see a Z disc. That's where you have thin fibers and then these cross-linking proteins. That's what Desmond is. So we've gotten through the cellular and basic tissue level anatomy of a skeletal muscle and cardiac muscle a little bit. Now let's talk about this process where an action potential or set of action potentials comes down a neuron and innervates several, usually it's several, sometimes it can be just a few, myofibers, that is to say skeletal muscle fibers. That axon plus all of the fibers it innervates is known as a motor unit. And where that axon contacts the skeletal fiber is known as a motor end plate. So you have an axon here. It's got vesicles full of acetylcholine. That's a neurotransmitter. And when those action potentials come, we release the neurotransmitter, and that then transfers. That initiates an action potential on the skeletal muscle. So let's dig into this a little bit. This process of linking one kind of cellular signal, that is to say a voltage type signal, that is an action potential, whether it's in the axon or on the skeletal muscle itself, to the response of contraction is known as excitation-contraction coupling. If we were just talking about pure cell biology, we'd be calling it signal transduction, but this is muscle, so it's excitation-contraction coupling. And it's a sequence of events. So what we have going on is that we get the action potential that is sent down by the axon, and when it hits the motor end plate, it will initiate an action potential on the surface and down the T-tubules. Those T-tubules are specialized plasma membrane structures that allow that action potential to spread evenly throughout the cell, all the way through it, all the way around it, so that the cell will contract as a uniform unit. And so that action potential spreads down through the T-tubules, triggering release of calcium from the sarcoplasmic reticulum. The sarcoplasmic reticulum is a calcium storage device first and foremost in skeletal muscle. That calcium gets released. The troponin will bind that calcium. It's a calcium sensor protein. And when it binds that calcium, it pulls tropomyosin away from those high affinity sites. And so now those black dots that you saw covered up in the previous picture are now available, and those myosin heads, those ATP metabolizing heads can now grab tightly that thick filament. We call that binding of the myosin head to the high affinity site of action a cross bridge. Okay. So the calcium binds troponin. That troponin then changes shape, pulling tropomyosin off of those high affinity sites, and those high affinity sites will now tightly bind myosin. A cross bridge is formed. And usually not just one cross bridge, there's a whole bunch of cross bridges formed. I gave you an overview in the previous slide. Let's kind of work through just the motor end plate, the neuromuscular junction, if you will. The motor end plate, neuromuscular junction, same thing. Okay. We get that action potential or series of action potentials coming to the axon terminal. This causes release of a neurotransmitter called acetylcholine. Better get to know that. That one is going to be your friend throughout neurobiology, Alzheimer's, you name it. Okay. Acetylcholine is released into that very, very, very short, thin space. These two cells are extremely close together. If you put your hands, like say you're trying to pray, you're kind of doing that classic prayer where you put your hands together. That is not as close as an axon and a skeletal muscle. It's very, very short distance. But that acetylcholine is released. It diffuses across. It's not very far. I mean, it's hardly, it's super close. So the diffusion is super fast, milliseconds. There are acetylcholine receptors on the surface of the skeletal muscle. And when they bind that acetylcholine, they will begin a process of depolarizing the skeletal muscle. Sodium flows in, the skeletal muscle voltage changes. We call that an end plate potential. And I don't know. I have mixed feelings about this, but I'm gonna follow the slide here. If you get just a teeny tiny bit of acetylcholine released, you'll get a little bit of depolarization. You will get a little blip on the voltage meter. Okay, that's an end plate potential. But if you have enough action potentials, you're gonna hit this thing called threshold. It's a voltage level where it triggers more action potential. Okay, I think this is an unnecessary complication. Okay, but it's in the slide, so I don't want to leave it behind. You get the beginning of a depolarization, and when you get more action potentials, you eventually reach this thing called a threshold, and that causes auto reinforcement of the action potential. Okay, so instead of it having to have the nerve continue to stimulate it, it now will trigger events on its own. Okay, and that's the wave of action potential that spreads all the way through the skeletal fiber. Now, you want this process to happen and end very quickly. You want this to happen, if you don't, you'll get tetanus. And so there's an enzyme that is sitting around in the space that actively degrades acetylcholine very, very rapidly. You want it to be super fast. You want contraction, relaxation, contraction, relaxation. If you have continuous, you want it to be voluntary continuous. You don't want it to be involuntary continuous. Okay? You ever sprayed a bug with Raid and just watch it? Okay? Sounds kind of morbid, right? Well, Raid is actually an acetylcholinesterase inhibitor for insect acetylcholinesterase. So what happens is you spray that Raid on that bug, it tries to contract its muscles, and the muscles can't stop contracting. Okay? The equivalent of Raid for human beings is something known as sarin gas. Nerve gas is what they call it. Okay? Does the same thing. In fact, certain types of insecticides that are acetylcholinesterase inhibitors, if you get a big enough whiff of it, you can actually begin getting symptoms of neuromuscular toxicity, just like the bugs. Okay, that's why there are certain types of insecticides which are banned because of that reason. Now, how does this relate to myasthenia gravis? Okay, so what myasthenia gravis is, it's an autoimmune disorder where the antibodies are attacking the acetylcholine receptors. And now they're binding the acetylcholine receptors, and you can't stimulate them with acetylcholine. So what do? You get muscle weakness. Okay? And you get breakdown of those acetylcholine receptors, which leads to muscle degradation. Okay, so we've done the excitation-contraction coupling. Now we've got that calcium out there. We're forming cross bridges. How do we actually generate the power? This is known as the power stroke. Okay, just like a rower when they go stroke, stroke, stroke. When they do a stroke, they're doing one pull. In this case, the power stroke is one pull of the catalytic head of that myosin. It's got a hinge region on it, and you can see here that it's sort of open, and here it's sort of closed. Okay, it's sort of flexed. Okay, we're going to kind of talk about that. Now, before we go any further, ATP hydrolysis, that is to say, turning ATP into ADP, is not the direct source of power. Okay? The ATP actually cocks the myosin into the correct position, and then when you form the cross bridge, the myosin is wound up like a spring from the ATP hydrolysis, and then it causes force on it. It is not the ATP hydrolysis that causes the force. Yes, the ATP, we're using the energy in the ATP in order to basically set the spring that is the myosin head. So let's walk through this a little bit. I'm going to do this visually first, and then we've got the narrative in the next slide. There's the calcium, binds troponin, and pulls the tropomyosin away from that site. We've got a cross bridge. Now, I want you to notice something. When that cross bridge forms, the ATP has already been hydrolyzed. Look at the angle of the myosin. You can see it's relatively open, especially down here. Look how closed that thing is down here. See that? It's way flexed. Think cocking as like take your elbow and bend your wrist towards your face. That is flexed. Now extend your elbow so that your arm is relatively straight. That's cocked. So ATP causes the myosin to go from flexed to cocked. And so in this first picture right here, we have ADP and inorganic phosphate. The ATP's already been hydrolyzed. It's already been cocked. Okay. The cross bridge forms, and that causes the cocking to switch to flexed. Okay. So when we bind when myosin binds ATP, it triggers flexion. In that process of flexion, we release ADP and inorganic phosphate. We actually release the inorganic phosphate first, ADP second. Okay. That causes the power. That's where the force is generated during the flex. Okay. We've lost ADP and inorganic phosphate, and here comes a new ATP in here. And guess what ATP actually does? It actually causes the myosin to release. Think about it yourself. Let's say you are pulling on a rope, hand over hand. You have to release a hand to reposition it, right? When you're grabbing that rope, you have to release one hand, you're pulling with the other one, you release one hand, you're pulling with the other one. That's kind of how myosin behaves. I think I'll try to find a video for you guys to kind of show this in class. When ATP binds, that causes the myosin to release the actin. The ATP then is hydrolyzed by the myosin, and that cocks the head, just as if you were moving your hand from below one wrist and putting it further up the rope. Okay? As long as those high affinity sites are available, we will cycle through this. Okay? So we get the calcium, it causes the opening of those high affinity sites. And then that causes a cross bridge formation, flex. When we flex, we release the inorganic phosphate and ADP. The ATP, the new ATP molecule comes along. This is a different one than this one. This is not the same thing. We got a new ATP comes in, causes the myosin to release, and it hydrolyzes the ATP in order to cock itself. And then, assuming that the high affinity site is available, we'll go through another one of these. All right. I know it's complicated. We will work slowly through this in class. Okay, let's bring the sequence together. This time it's in a narrative form. Okay, so listen closely, write a word or two down just to kind of remind yourself. Don't have to write everything down because then you got to read everything again. Short phrases, things that remind you. Okay, so the first thing, we get calcium that's released. That's the excitation-contraction coupling. Binds to troponin, and troponin moves to tropomyosin and uncovers that high affinity active site on actin. It's really a high affinity site. It's not active. Okay? Myosin head binds that high affinity site, forms that cross bridge. This binding and forming a cross bridge initiates the power stroke. The myosin head has already been cocked before this. So what happens is we bind, and then the first thing we release is the inorganic phosphate, and we are flexing, we are flexing, we are flexing. Okay, that's where we're generating force on the actin. As we approach the end of that flex, we also release ADP. So by the end of that flex, we have released inorganic phosphate and we've released ADP. A new ATP comes along and attaches to the myosin head, causing the release of the myosin so that it can reposition. And what it does is it hydrolyzes that ATP into ADP and PI. Okay, that's the hydrolysis of ATP into inorganic phosphate and ADP. It's no longer attached to each other. That cocks the myosin. It repositions the myosin head. And if assuming that the troponin and the tropomyosin are still activated by calcium, we would repeat by step 2. If the calcium has gone away, we would stop here basically at step 0. Okay, step 0 would be where the troponin and tropomyosin are covering the high affinity site. But if there's still calcium available, the myosin will basically repeat step 2. through step 6. Let's take some of the microanatomy. I know that I didn't talk too much about H zones and A bands and that sort of thing. So I'm going to pick it up here. Okay. And again, just try to, work a little bit here. The main thing that happens when we have these sliding filaments is that the 2 Z discs, okay, by the way, the I band is all actin, and the Z disc are the desmin and nebulin and some other things that link thin filaments together. So there's a Z disc and another Z disc. That defines one sarcomere, a Z disc to Z disc. Okay, the I band is actin only. The A band is overlapped actin and thick filaments. So thin filaments and thin filaments overlapped. There is this lighter zone that's only thick filaments. You see there's no thin filaments here. That's called the H zone. And so what happens is we know it's a sliding filament, first of all, because the Z discs get closer together. And notice that the I band all but disappears. Okay, this one isn't contracting over here. Somehow they were able to get this one sarcomere to do it. Okay, notice that the I band is much, much thinner, and the H zone all but disappears. That's because now we have complete overlap of the actin and myosin. Okay. Just a little bit of I band left, but you see the Z discs are closer together and the H zone has disappeared. But the A band hasn't. The A band is still pretty much the same size. There used to be a competing theory about myosin that would shorten rather than the sliding filament. And so if the myosin was shortening, what you'd see is the A band getting skinnier, but it doesn't. Okay? What you see is the I band disappearing and the H zone disappearing. That means that the actin is being pulled into the A band. You might remember that with skeletal muscle, you kind of can't keep it contracted for very long. Okay. Most times it's usually pretty just one and done, maybe one every few seconds or one every second, sometimes a little faster, but not all that often. Okay. Well, that means we have to have almost immediate relaxation. Now, if you keep stimulating, you keep releasing the acetylcholine, you'll get tetanus. That's true. But most of the time, we aren't doing super long contractions, a few seconds, all right? So we need a relatively rapid way to relax. Otherwise, we'll go into tetanus every time we try to contract a muscle, okay? So releasing that acetylcholine, well, one way is to get rid of the acetylcholinesterase really, really quickly. You don't want to have this stuff sitting around. Like I said, if you have an inhibitor of acetylcholinesterase, that can lead to uncontrolled tetanus, seizures, and eventual muscle failure, like in RAID, okay? So we're going to break down that acetylcholine really, really quickly, okay? We're also going to be pumping calcium back in really, really fast, okay? So we're pumping the calcium back into the sarcoplasmic reticulum using ATP, by the way. This is why you don't want to run out of ATP. It's not a good thing, okay? As long as calcium is out here, we're going to get cross bridges forming because the tropin myosin is revealing the high affinity site. So as long as calcium stays here, we're going to cycle through the power stroke, okay? So we get, so we're going to remove the acetylcholine, prevent additional depolarization of the cell membrane of the skeletal fiber. We're going to use active transport, that is to use ATP to pump calcium back into the sarcoplasmic reticulum. So whatever calcium has been released, we are pumping it right back in. So get rid of the calcium. And when you get rid of the calcium, the tropomyosin will slide back over those high affinity sites, and so the myosin can't bind the actin. Now, when myosin is binding ATP, it has to release actin. And so that will allow the tropomyosin to slide in if there's no calcium around. So as we release the actin during that ATP binding and subsequent hydrolysis, that's when tropomyosin, if there's no calcium around, will slide back over that high affinity site. And what happens is the myosin can no longer associate with the actin, so you don't generate force anymore, and so the sarcomere returns to its original resting length. Now, what's this here? Rigor mortis. I know we've all heard of it. Sometimes when you have a body that's been dead for not very long, they call it a stiff, right? Well, the rigor mortis and the word stiff are closely related. Rigor mortis means the stiffness of the rigidity of the dead, okay? The rigidity of death. So if the skeletal muscle runs out of ATP, like when it's dead, what will happen is it will flex. Now remember, when you flex, you release ADP and inorganic phosphate. But if there's no ATP here, it's basically stuck. The ATP is required for that muscle to relax, okay? Because what happens is we release the actin, the ATP is hydrolyzed, and we reposition the myosin. The only reason we don't go through another power stroke again is if there's no calcium there, the troponin and tropomyosin slide over that high binding site and you can't form a cross bridge. Okay? But if you don't have ATP, you are stuck in what is known as the rigor state. You are flexed and you are cross-bridged. You are tightly bound to actin. That's because there is no ATP to allow first release and repositioning of the cocking, if you will, of the myosin head. And this will last for several hours. So after a body dies, beginning about, I don't know, a few minutes after death, it'll be more than a few minutes. Let's say 30 minutes, okay? 30 minutes after death up to about, 12 or 18 hours. I forget exactly. In fact, they can time it pretty well. If you're into forensics, they time that stuff like crazy, okay? They know that if the body is stiff, it's died within a certain period of time, and then there are other signs like lividity and other things along those lines. But rigor mortis is the rigidity of the dead, and that's due to basically due to ATP depletion. You're stuck in what is called the rigor state, where you are flexed and tightly bound, okay, and you don't have ATP to cause release and reposition that myosin. Okay, I'm going to stop right here for now. We've been running for about 50 minutes. I know it's been going a little while, a lot going on there. Something I want to advise you on, and I know this isn't advice you necessarily want to hear, but the key to this stuff is investing quality time. This is not part-time stuff, okay? I know that we're on the fast track, but that means more time more often is what it means. There's no less time here. And so please do take your time with this material. Please ask questions. Let's work through, try to understand it as best we can in the time that we have. I will be creating a part 2 with the rest of the slides. I hope you've enjoyed this, and please, by all means, let me know if there's anything I can do to help you. Talk to you soon.